Chapter 10, Section 4, Meiosis. Learning objectives. Identify how many sets of genes are found in most adult organisms. Describe the events that occur during each phase of meiosis. Identify the differences between meiosis and mitosis. Explain how two alleles from different genes can be inherited together. Each cell of the fruit fly, Drosophila, has 8 chromosomes. 4 of these chromosomes came from its male parent and 4 came from its female parent. These 2 sets of chromosomes are called homologous, meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. A cell with 2 sets of homologous chromosomes is said to be diploid, meaning double. The diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. The diploid number of chromosomes is sometimes represented by the symbol 2n. Thus, for the Drosophila, the diploid number is 8, or 2n equals 8, where n represents the single set of chromosomes found in the sperm or egg cell. Some cells have a single set of chromosomes and therefore a single set of genes. Such cells are haploid, meaning single. The gametes of sexually reproduced organisms, including fruit flies and peas, are haploid. Remember that mitosis consists of 4 main phases: prophase, metaphase, anaphase, and telophase. And at the end, you have an identical set of 2 daughter cells. How do sexually reproducing organisms produce haploid, or N, gamete cells from a diploid, or 2N, cell? That's where meiosis comes in. Meiosis is the process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in the diploid cell. Meiosis usually involves 2 distinct divisions called meiosis I and meiosis II. By the end of meiosis, a single diploid cell has produced 4 haploid cells. Just prior to meiosis I, the cell undergoes a round of chromosome replication during interphase. Each replicated chromosome from the male and from the female consists of 2 identical chromosomes joined at the center. After interphase I, the cell begins to divide and the chromosomes pair up. In prophase I of meiosis, each replicated chromosome pairs with its corresponding homologous chromosome. This pairing forms a structure called a tetrad, which consists of 4 chromatids. As the chromosomes pair, they sometimes undergo a process called crossing over, in which bits and pieces of homologous chromosomes are exchanged. Crossing over produces new combination of alleles on each chromosome. As prophase I ends, a spindle forms and attaches to each tetrad. During metaphase I of meiosis, paired homologous chromosomes line up across the center of the cell. As the cell moves into anaphase I, the homologous pairs of chromosomes separate. During anaphase I, spindle fibers pull each homologous chromosome pair towards opposite ends of the cell. When anaphase I is complete, the separated chromosomes cluster at opposite ends of the cell. The next phase is telophase I, in which a nuclear membrane forms around each cluster of chromosomes. Cytokinesis follows, forming 2 new cells. Meiosis I produces 2 daughter cells. However, because each pair of homologous chromosomes was separated, neither cell has the 2 complete sets of chromosomes in a diploid cell. Those 2 sets have been shuffled and sorted almost like a deck of cards. The 2 cells produced by meiosis I have sets of chromosomes and alleles that differ from each other and from the haploid cell that entered meiosis I. This is due to crossing over. The two cells now enter a second meiotic division, called meiosis II. Unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis II. As the cells enter prophase II, their chromosomes, each consisting of two chromatids, become visible. The chromosomes do not pair to form tetrads because the homologous pairs are already separated during meiosis I. During metaphase of meiosis II, chromosomes line up in the center of each cell. As the cell enters anaphase, the paired chromatids separate. The final four phases of meiosis II are similar to those in meiosis I. However, the result is 4 haploid daughter cells. In the example shown on the picture here on the slide, each of the 4 daughter cells produced in meiosis II receives 2 chromosomes. These 4 daughter cells now contain a haploid number, or N, just 2 chromosomes each. The haploid cells produced in meiosis develop into gametes for sexual reproduction. In males, these gametes are usually called sperm. In females, they are known as egg cells. Remember that in meiosis, it consists of 2 main phases, meiosis I and meiosis II. Crossing over occurs in meiosis I, and the result is 2 daughter cells. In meiosis II, chromosomes do not replicate prior to prophase II, and the result is 4 haploid daughter cells. The words mitosis and meiosis may sound similar, but the two processes are very different. Mitosis can be a form of asexual reproduction, whereas meiosis is an early step in sexual reproduction. Mitosis and meiosis also differ in a way chromosomes move and in their number of cell divisions. A cell replicates or copies all of its chromosomes before entering either mitosis or meiosis. However, the next steps differ dramatically. In mitosis, each daughter cell receives a complete diploid set of chromosomes. In meiosis, homologous chromosomes are separated and each daughter cell receives only a haploid set of chromosomes. As a result, in meiosis, the 2 alleles for each gene are segregated and end up in different gamete cells. The sorting and recombination of genes that takes place in meiosis helps to increase genetic variation from one generation to the next. This also explains why you do not look identical to your parents. Mitosis does not change the chromosome number of the original cell. Meiosis reduces the chromosome number in half. Mitosis also results in the production of 2 genetically identical diploid cells, whereas meiosis produces 4 genetically different haploid cells. Thomas Hunt Morgan first realized in 1910 that he was curious how genes were inherited together, and he realized that this might have something to do with gene linkage. Morgan's research on the Drosophila fly led him to the principle of gene linkage. Morgan identified more than 50 genes and the traits they caused. He discovered that many of the genes appeared to be linked together in ways that, at first glance, seemed to violate the principle of independent assortment. Morgan's findings led to 2 remarkable conclusions. First, each chromosome is actually a group of linked genes. Second, Mendel's principle of independent assortment still holds true. It is the chromosome, however, that assorts independently, not individual genes. Alleles of different genes tend to be inherited together from one generation to the next, when those genes are located on the same chromosome. In 1911, a Columbia University student was working part-time in Morgan's lab. This student, Alfred Stuyvesant, wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's locations. Stuyvesant reasoned that the farther apart 2 genes were on a chromosome, the more likely it would be that crossing over would occur between them. If 2 genes are close together, then crossing overs between them should be rare. If two genes are far apart, then crossing overs between them should be more common, reducing the frequency in which they are linked. By this reasoning, he could use the frequency of crossing over between genes to determine their distance from each other. Stuyvesant gathered up several notebooks of lab data and took them back to his room. The next morning, he presented Morgan with a gene map showing the relative locations of each gene on one of the Drosophila flies and their chromosomes. Stuyvesant's method has been used to construct gene maps. A gene map shows the location of a variety of genes on chromosomes.